Good afternoon to everyone. Okay, and in this talk I will try to make you taste the flavor of the activity we are carrying out in our institute, which is the Institute of Applied Physics, within clearly the Spoke 4 activity. And so I will speak mainly about two things. One is associated to the use of optical nanostructures to be internalized inside cells. And then I will speak shortly about the optical platforms which are developed for the detection of biomarkers of interest in the lung and or eye diseases. And more specifically, I will speak about molecular beacons for theranostic purposes. And I will speak about two different platforms, one based on the use of surface enhanced Raman spectroscopy, and the other one based on long period gratings. So briefly, just an introduction of what a molecular beacon is. A molecular beacon is a sequence of oligonucleotides, which in a normal situation is in a close conformation here with a quencher and a fluorophore, which are at a short distance. So what you have darkness in principle. But if you have I can do it, okay. If you have the target, the structure opens, the fluorophore is far, more than 100 nanometers far from the quencher, and the fluorescence resonance energy transfer does not occur, and you have emission of fluorescence. So you can see with, let's say with eyes, the presence of the target. And this can be used by the choice of the target, which generally can be, for example, RNA messenger or microRNA, in any case, a sequence of oligonucleotides, and to characterize the sensor constituted by this molecular beacon. And you can apply this either inside the cell to internalize this molecule inside the cell, looking at some target, for example, in our case, mRNA messenger, which are related to particular pathologies. And so we are able to detect RNA messenger inside the cells in a certain point. And we can study the fate of the molecular beacon and the carrier, because clearly, as all of you know, I think that clearly this molecular beacon does not enter inside the cell by itself, clearly. Or we can use out of cell, immobilizing this molecular beacon on a suitable surface, and to have so a sensing probe outside of the body, clearly, to make a sensor, a biosensor. And in this case, we use for the detection of microRNA. So I will speak about the targets and aims within Spoke 4 for a moment, which are concentrated on detection of the mRNA, which is associated to the production of salvivin, which is important in cancer. And we will see how this sensing probe can be used as a theranostic element. And we concentrate with optical platforms for the moment on microRNA 183, which is expressed in chronic obstructive pulmonary disease, COPD. So let's go to the RNA messenger for salvivin. So salvivin belongs to a family of proteins which are called as inhibitor of apoptosis, and it's clearly important in the role of cell duplications and proliferation. And it's quite evident that salvivin expression is very high in cancer, in many cancers where there is a proliferation of cells, because there is an overproduction of this protein. And this is quite old table where we see that there is high overexpression of survivin in practically almost all, the great many of the cancers. So what we can do with our molecular beacon, supposing to be able to internalize inside a cell. So what you have, if this molecular beacon enters inside melanoma cells, this probe meets the RNA messenger for the product, which is associated to the production of survivin, binds, and if it binds in the right position, stop the production of survivin. So clearly, if in normal cells there is no RNA messenger for survivin, or a very low amount, you do not have any optical cinema. But the important aspect is that what we have, if we design our molecular beacon in order to bind in the region of the RNA messenger, which is coded for the production of survivin, the production of survivin is stopped or is decreased. And this clearly has a pharmacological effect, and it was demonstrated in our study. We, at the beginning, we started, I will speak about after. So the problem is this one. How can we make enter the molecular beacon inside? Because molecular beacon is negatively charged. The surface, external surface of the cell is negatively charged. So clearly it's a protection for the cell that no DNA sequence enters, because can alter completely the environment of the intracellular environment. What we need is to develop or to use cargos, vectors, which are designed in order to be able to carry on with them the molecular beacon. And there is an infinity of, okay, well, I stop to speak about the possibility of these vectors because the literature is full. And the idea is to have clearly something which has to be biocompatible, has to be able in case to recognize with the target molecule the cancer cell and so on. So this is the activity where we are. We made a lot of study using PMMA nanoparticles, with clearly are synthetic nanoparticles. They are not satisfactory. And within this project, we started a collaboration with Professor Agnese Magnani, who is here, for the encapsulation on for us, a novel type of intracellular vehicle, which are lipid-based nanoparticles. This encapsulation was successful, and now we are going on making all the tests on adenocarcinoma A549 cells. As I told before, that we are also active in the development of platforms, and we use in this case a molecular beacon which is designed for microRNA 183, which is a microRNA circulating and is specific for the presence of COPD disease. I forgot to tell that the molecular beacon we use for the survivin was already described in literature, not like molecular beacon, but like silencing RNA. But now the so-called CNR now are very popular due to the Nobel Prize, which was given a few days ago on the scientists who started this type of activity, starting the study on mRNA messenger and so on. In this case, we design, it's possible to design the molecular beacon knowing clearly the sequence of these microRNA oligonucleotides. And this is the sequence that we designed, and this is the closed conformation, and this is the open conformation in the presence of the complementary target. we tested this this molecular beacon, and we couple this molecular beacon on an optical platform working on surface-enhanced Raman scattering. How it is work? When I told before you there was about the previous molecular beacon, there was a fluorophore and a quench red distal lens. In our case, we have a Raman reporter, and the other side is fixed on the surface, on let's say a surface which is sensitive to SERS, constituted by silver nanowires. And so how the modulation of the signals occurs. In this case, what you have in a closed configuration, you have that the Raman reporter is close to the nanowires, so you get a very high cell signal. In the presence on the target, the distance increase, and you see a decrease of the Raman spectrum. And this is the working principle by which we can use this molecule, nanostructure, to create a platform based on surface-enhanced Raman scattering. So this is the fabrication of the SERS substrate, which is constituted by a layer of silver nanowires. By laser engraving, you get 4 different spots, and these spots are included in a particular flow cell. I don't go into details clearly on this, but you have here, there are the 4 spots with these 4 walls, which are interrogated by means of a Raman spectrometer, and you have the change of the conformational change of the molecular beacon as I showed before. And in this case, we are quite far with the research, because we, as I told you, we have a decrease of the Raman spectrum with the target increase, target concentration increase, and we checked that the fact that there is a good reproducibility of the Raman spectrum which is collected on the spot, because clearly the spot has quite large dimensions, so you can collect the spectrum in different, close but far in terms of the probe for the interrogation of the spectrum. And we got a quite sensitive calibration curve where we are able to detect with a limit of detection of the order of few tenths of femtomolars. Sorry. And it is quite selective, sorry. With a single mismatch, we have not a so large signal, and is reversible, because if we are now out of the body, we want to use our sensor more time, that we have to regenerate the surface. And so that's quite good results. And in the last 2 minutes, the second photonic platform that we are developing based on, in this case, the use of long period grating structure, where you have a modulation of the refractive index inside the fiber, which gives you the possibility to launch a signal and to have a modification of your signal, which is transmitted through the fiber. And in this case, long period gratings means long because the modulation, the pitch of this structure is quite long, in order that part of the radiation, sorry, okay. I pushed too many buttons. Okay, thanks. Okay, what you have is that if the pitch is so large, part of the radiation goes outside of the fiber and sees the external environment, sees the change of refractive index. So you can do a changing the refractive index of the external medium. You have a shift of these peaks, which are the resonance peaks, which are created by this modulation of the refractive index inside the fiber. You change the refractive index, more dense, and you have a further movement of this. And so you can measure with this. But okay, this is clearly a measurement of the refractive index of the solution. So how can I do with this approach a biosensor? The idea is that if you have a bio layer with, for example, a receptor, a biological recognition element, let's say an antibody, this creates a change of refractive index, clearly small, but this is a very sensitive, capable to detect 10 to the minus 4, 10 to the minus 5 change in refractive index, and you have so a shift of the peak due to the presence of the antibody, and if you have the captured antibody, or you have an antigen and you have a sandwich layer assay, you have this shift in the resonance peak that you can detect. The important thing is that on the same fiber, you can have more than one grating, characterized by different pitches, characterized by different resonance wavelength. And so you can measure with a single fiber more than one analyte. And this is the activity that we are developing within this, the spoke for the idea to have microfluidic cell. You have clearly with the fiber here in this groove. Clearly you have everything which is sealed, and you make the measurement, and you have, in this case, a microfluidic circuit where you have practically, this is all the fiber. In this area, you have a single grating, in the other, another LPG grating, and you have microfluidic which runs in this way, which allow you to immobilize something on one grating, and then you can do the same for the other part. And so you have, along the same fiber, two different biolayers capable to see different molecules, and the measurement is done clearly, in this case, all along the fiber. And now clearly this is coupled with a peristaltic pump, and we want to develop a microfluidic with a syringe pump, and creating something which is more compact and, let's say, transportable like a POCT device to measure. At the moment, we are concentrating on the protein FKBP12 in collaboration with Professor Caminati of the University of Florence, who complete the synthesis of a receptor molecule, which has to be mobilized on the fiber, and then capable to detect this protein, which is involved in lung cancer. And then we can also immobilize and detect on the other, with the other grating, tacrolimus, which is another protein which is associated with this. And so with this I finish. I showed you what we are doing for intracellular tests using molecular beacon in collaboration with Siena, and the development of the cells-based optical platform, which is almost at the final stage. And we have to thank clearly the spoke for, because, for example, the collaboration with Gabriella Caminati and Agnese Magnani started within this project. And clearly, these are the actors of this activity. We are the two groups working at the Institute of Applied Physics, which is our group, which is Chem and Biochem Optical Sensor Lab, and the other one with Paolo Martini, Roberto Pini, which was the director of our institute, which is Biophotonics and Nanomedicine Lab. So we are very, very correlated groups. And that's all. I hope to have been on time in the question. So you can close, okay?